So welcome everyone. I'm going to present this webinar on the deep learning imputation for peptide bioactivity and property prediction. I'm going to cover a few topics. First, I'm going to give a short introduction onto the challenges in peptide discovery and our collaborative project with MSD. I'm going to talk about our platform Cerella, which provides a robust and accessible deployment to guide drug discovery, and how we applied Cerella to address some of the challenges in peptide modeling. And I'm going to cover how we generated reliable predictions for peptides, activities, and properties, how we view Cerella to select the best peptides according to the desired criteria, and how it can provide insights into peptide optimization and guide experimental design. And then I'm going to wrap up with some conclusions. So to start with an introduction on the challenges that we can encounter in peptide discovery, we know that machine learning methods have been used in the drug discovery projects for small molecules to improve efficiency and productivity in these projects. And for these machine learning methods to be successful, we need to rely on good representation of the molecular structures. However, when it comes to peptides, because their activities and properties rely on their flexibility and conformations, these properties cannot be captured very well using small molecule descriptors, which makes the prediction of activities and properties of peptide drugs even more challenging. And so that's why conventional machine learning methods can struggle to predict activity values for peptides prospectively. And so this is an example here for a peptide, tachylepsin, one which has three conformations. So the structure and flexibility can change for those peptides. Another challenge that we can encounter in the drug discovery of peptides is how expensive and time-consuming running the experiments to generate peptide activity and property profiles. And so because these experiments are very expensive, we would expect that the experimental data for peptides are quite sparse, which makes finding and identifying peptides with the optimal balance of properties is quite challenging when this data is quite missing. And so it's very important to prioritize experimental tests, which would give us the most information with which we can make decisions. So in this presentation, I'm going to cover our collaboration with MSD, where we collaborated on a project to model peptides using two of their property data sets of peptidic compounds. They are mostly microcycles of 6 to 15 monomers. And so the two projects that we've explored, which are being named here as Project 1 and Project 2. Project 1 had 33 experimental endpoints and over 5,000 peptides. Project 2 had 39 experimental endpoints and over 1,000 peptides. And so we've applied this Cerella platform to model peptides in these data sets and compared that with multiple methods, including random forest, chemprop, and KNA and imputation. We've also explored a wide variety of descriptors, including 2D startup descriptors and sequence-based descriptors. So I'm going to introduce the Cerella platform. So what is Cerella? Cerella is a proven and deployable AI platform that empowers drug discovery teams to go from concept to candidate faster by using early stage data so that we can confidently predict late stage expensive outcomes. It can help pinpoint better compounds for your projects by identifying hidden opportunities that are caused by missing, uncertain, or incorrect data. So we can comprehensively explore the optimization strategies to prioritize compounds for synthesis and testing. It can also inform decisions by giving insights regarding to the mechanism of actions and adverse outcome pathways. So it's an interpretable platform. And it can also help choose the most valuable experiments to guide compound progression. So what does that mean in the context of reducing time and cost in drug discovery cycles? So Cerella can help highlight high-quality compounds proactively by filling the gaps in the sparse data using imputation. It can help increase confidence in decision-making by identifying hidden opportunities, flagging outliers, and false negatives. We can also translate the insights from the Cerella platform into planning experiments and focus on the most valuable measurements to make. We can also gain more value from the data by accurately predicting complex endpoint, overcoming performances from conventional QSAR modeling. And because it also provides interpretable insight, it can identify many of the nonlinear correlations, even if the data is sparse, which allows us to understand mechanism of actions and adverse outcome pathways. So we've applied Cerella in the past on many small molecule projects in collaboration with many of our partners in the pharmaceutical and biotech field at different stages of the drug discovery process, from hit to lead optimization, and preclinical development. And we've shown a track record of success, even in non-pharma applications, such as imputing in vivo sensory properties and predicting agrochemical bioactivity profiles. So the question that arises, can it work with peptides? Can Cerella help guiding peptide drug discovery? So the first application that I'm going to cover is how Cerella can help generate reliable predictions for peptides. So I've already discussed how conventional QSAR methods can struggle to make predictions for peptides because their peptides, activities, and properties rely on their conformations, which might not be captured very well using small molecule descriptors. Another limitation in the peptide modeling is the high experimentation cost and consequently data sparsity, which can be a block for building robust QSAR and deep learning methods. So the question, how can we use Cerella to help address these limitations of molecular descriptors and the challenge of sparse and noisy experimental data for peptides? So before I describe the engine, the deep learning imputation engine behind Cerella, I'm just going to make a distinction between prediction and imputation. While we understand the term prediction, which uses features such as molecular descriptors as input to predict one or more property values for a compound, such as a random forest in this case, the imputation method is the process of filling the gaps in a sparse experimental data matrix using the limited data available to fill in the gaps and produce a fully imputed experimental data matrix. And the deep learning imputation method that behind Cerella is called Alchemite, which is developed by our partners at Intelligence. And the key advantage of the Alchemite method is that it learns directly the relationship between experimental endpoint in addition to the structure-activity relationship. So in a matrix, it would take both the descriptors and the sparse experimental data as input to fill in the gaps in the matrix, generating predictions that are more accurate than conventional QSAR methods, so it captures more informative patterns in the data. And so by filling in the gaps in the data matrix, we can quickly highlight and target high-quality peptides. And as a bonus, the Alchemite method can also generate high-quality predictions for compounds that have not been tested, so using only molecular descriptors as input, where we can prioritize in this way new designs for synthesis and testing. So before going into this, describing the results, I'm just going to quickly show how we assess results for a data set with many endpoints. We use the coefficient of determination, the R-squares as a metric of accuracy, where the higher the value, the more accurate the results. And we calculate the coefficient of determination on the test set for each of the endpoints in the data set. We can plot that in a performance profile where we have the coefficient of determination, the R-squared in the y-axis, and the endpoints in the x-axis ranked by and ordered by accuracy. So each of the points represents a result for a single endpoint. And as that line goes towards the top right, it shows an increasing overall accuracy. We can capture or summarize the overall accuracy by reporting a median R-squared for the results of a given model. So we are going to demonstrate results on two projects, so Project 1 and Project 2. And I'm going to start with the results for Project 1, where I'm comparing the alchemite imputation results with other methods that we've used. We've compared the alchemite imputation with random forest KNN imputation and chemprop. The alchemite imputation here is displayed in the blue line. And what we can see from this profile is that separation in the lines between the imputation method and other methods, where we see that the performance by imputation is statistically different, despite it's a small difference, but it's significant difference compared to QSAR methods. We also noticed that there was insignificant difference between the virtual and QSAR methods. So when we don't supply the alchemite with experimental data, it performs similarly to conventional QSAR methods. We know that a subset of these endpoints have the most valuable, and are the most expensive ones. And so we can focus on looking into the results on that subset. What we've seen that we achieved excellent performance, our R-squares greater than 0.7 on almost all of these endpoints. However, we don't see substantial improvement from imputation for these endpoints. When we look at Project 2 results, here we can see actually a statistically significant and substantial difference between imputation and other methods. So we see that big separation between the lines in the performance profile. However, when we compare the virtual and QSAR methods, we don't see significant difference in performance. By focusing on the subset of the most valuable endpoints, we again can see that substantial improvement from imputation for these endpoints. And the reason for that is that the imputation method leveraged the relationship between endpoints to make accurate predictions, even when the data is sparse. And so that means that we can overcome challenges in peptide modeling by making a better use of limited data in a way that is outperforming conventional QSAR methods and hence generating more accurate predictions. So now we've demonstrated that we can generate more accurate predictions using the imputation method. Let's see how we can use these accurate predictions to select the best peptides according to a set of desired criteria. In order to find an optimal balance of property within peptides, we need to have more data. And since generating experimental data for peptides is quite challenging because it's time consuming and expensive, most peptide data is sparse, which makes that achieving of the best peptides with the optimal balance of properties quite challenging. So we've demonstrated how the imputation methods can actually fill in the gaps in the sparse experimental data. So how we can use this richer data to identify the best peptides. So the process of finding the optimal balance of properties, which we call the multi-parameter optimization in drug discovery, we would like to find the hit candidate that would achieve a good balance of different properties. However, we would like also to quickly identify a situation when this balance is not possible, so that we can reduce experimentation costs and also only focus when we have confident results. One way to achieve that is through probabilistic scoring in our software StarDrop, which evaluates all available data against project criteria. It accounts for uncertainty in the compound-related data, and it provides an objective assessment of compounds' chances and likelihood of success. So the expert user would define how a good compound look like by defining the scoring profile and identifying which properties are important at which levels and ranges of values, and then the probabilistic scoring functionality in StarDrop will calculate a score that reflects the likelihood of success. We can rank these compounds based on that score, and the compounds that have the highest likelihood of success would float into the top. And the visualization of that score includes histograms for visual guides for the different compound properties and their contributions towards the score. So in order to calculate the probabilistic scoring, we would need three key ingredients. One is the property data, whether it's generated from experimentation or predicted values. Second is the criteria for success, how a good compound look like. And thirdly, the uncertainty in the data, whether that is in experimental or predicted value. And by using these three ingredients, we can calculate a score which reflects the likelihood of success and an accompanied confidence score. What we can do with the results is visualize compounds based on their rankings, where we can quickly identify compounds that are likely to meet the desired criteria and reject with confidence compounds that are likely not to meet the criteria. So we've demonstrated that the imputation method can generate accurate results where the data is missing. We can define the criteria for how good peptides look like. Let's examine how we can generate robust uncertainties in predicted data. So one of the key advantages of the outcome methods, which is the deep imputation method behind Cerella, is that it generates an estimated uncertainty for each individual prediction. So instead of generating a single prediction, it would generate a distribution of predictions, which the width of that distribution reflects the confidence of the model. And what we've seen over and over, the strong correlation between the uncertainty estimates and the observed accuracy on independent test sets. And so by focusing on where these results are confident, we can highlight the most accurate prediction with which we can base our decisions. So how we can examine and test and assess the relationship between the estimated uncertainty and accuracy. So I'm going to use one example for one of the peptide activity endpoints, where it's shown in the right-hand side here, where we can look at the scatter plot of the observed versus predicted values. So each of the points represent that relationship between observed and predicted value, and these error bars reflect the uncertainty of the model on that prediction. So while overall we can see a good correlation between observed and predicted value, some of the points are off the identity line. We can assess the uncertainties by looking at the RMSE, the root mean squared error, where we can plot that against the ranked order of predictions. So in the left side plot, we can plot the RMSE, which increasing in accuracy from top to bottom against the predictions ranked by accuracy. So for 100% of predictions, the RMSE of the points overall is around 0.4. What we can do is eliminate and remove predictions with the highest uncertainties, focusing on the most accurate predictions. We would find that points become closer to the identity line. They have smaller error bars, so these are the most confident results. And that is reflected in a lower RMSE value. So the model is providing more accurate results as we focus on the most confident predictions. We can go further and focus on the most confident 10% of predictions. And here we see how the points become even closer to the identity line, reflected in the plot to the left, with a lower value of RMSE for 10% of the most confident 10% of predictions. And this demonstrates the high correlation between the model estimated uncertainty or confidence and the observed accuracy. And this means that we can rely reliably, we can rely on the predictions to focus on the best peptides with confidence. So now we've ticked the third box in our ingredient for calculating the probabilistic scoring. We can use these three elements to calculate a score to rank peptides for their likelihood of meeting the desired criteria. So as a result is a profile where we have peptides ranked by their scores. The high quality peptides are ranked to the top, which are easily identified. And so we can conclude that by combining accurate predictions from imputation, robust estimated uncertainties, and probabilistic scoring, we have higher chances of identifying peptides of interest with the highest quality. So we've demonstrated how the imputation in Cerella can help generate reliable predictions, how it can be used to identify peptides that meet the desired criteria. Let's see how it can provide insights for peptide design optimization and guiding experimentation. So in order to optimize peptide structure, we need to have a good understanding of the structure-activity relationship. And in order to achieve that, we need to relate the information from predictive models to the characteristic of individual monomers in the peptide sequence. We can also appreciate that that generating activity and property profiles are time-consuming and expensive. So it would add so much value if we can identify those experiments that will provide the most information with which we can choose our best compounds. And so what I'm going to demonstrate next is how we can identify monomer positions and properties in the peptide sequence alignment related to the endpoints of interest, and how we can identify the most important assay to run with which to accurately impute expensive late-stage experiments. So to gain insights into the structure-activity relationship, we need to incorporate interpretable descriptors. And in the context of peptides, we can describe the peptides after performing sequence alignment, where we described each monomer in the alignment by its position. And for each monomer in the alignment, we can describe that with a vector of interpretable monomer descriptors that reflect, for example, log P, flexibility, and aromaticity. And by doing that for each monomer in the alignment, we generate sequence-based descriptors that are interpretable and used to supplement imputation models. And in this way, we can infer insights about important monomer properties and at which positions. And the way we use that is by supplementing the imputation model by whole molecule descriptors, sequence-based descriptors, and experimental data. So in a multi-domain fashion, where the imputation model will learn the important experimentation and the important experimental test, as well as the peptide, molecular, and sequence-based properties for the endpoints of interest. So how we can retrieve these insights? So the Alchemite methods behind Cerella can generate interpretable relationships between experimental endpoints and descriptors. And in this way, we can gain insights into structure-activity relationship, mode of action, adverse outcome pathways. So by generating this importance matrix, which describes the relationships between each of the inputs, whether it's a descriptor, molecular descriptor, sequence-based descriptor, or experimental endpoint for each of the outputs. So the higher the magnitude of that value, it reflects the strength of the importance of that input for the output of interest. And here, the higher the value is highlighted in a higher color of red, higher intensity in the color. And I will show later how we can interpret those matrices. So by first looking at the results from project 1, and as we've seen previously in this project, we have observed small improvements on the overall performance for imputation over QSAR and virtual methods. And by looking at the importance matrix profile for inputs versus outputs, so the inputs in this case are the descriptors, whether they are from molecular descriptors or sequence-based descriptors, which are highlighted in blue, versus inputs from the experimental endpoint space. So we can generally see how many of these red squares are highlighted within the descriptor space. If we examined one of the endpoints of interest, in this case, we can display the importance of the most informative experiments for that endpoint. And what we are seeing here is small importance values are observed for the informative experimental endpoints. And this is consistent with what we've seen in before, where we have small improvements seen of imputation over a virtual model. And so the reason for that is that the predictions in this project are derived by structure-activity relationships, which are captured by structure and sequence-based descriptors. And so, which means we can gain information from some of these descriptors and how they relate to endpoints of interest. So for example, for an endpoint, which I will call here X, that was influenced by the number of aromatic atoms at position 4 in the peptide alignment. In another example, for an endpoint, which I'll call Y, that was informed by the monomer log P at position 12 in the sequence alignment. So we can see how these insights and how these interpretable relationships can provide guidance into the monomer positions and properties, which we can focus for peptide optimization. So let's look at the results for project 2, where we've observed large improvement in overall performance for imputation over virtual. And by just looking at the importance matrix, we would see that most of the intense red color is within inputs that are from the experimental endpoint space. If we examined one of the examples of the most valuable endpoints, we can see that the importance bars for the most informative 5 experiments are higher. So the high importance of these experimental endpoints corresponds to the substantial improvement that we've seen for the imputation in this project. And this translates into informing which moderate cost experiments that can be valuable to impute high value outcomes. So how Cerella can help in terms of identifying informative measurements. So we've demonstrated how these relationships can highlight the most informative experiment to test, but Cerella can provide even more information into identifying which measurements to make. So not only which endpoints, but also which compounds that we would like to focus on, so that we can impute a broad range of activities and properties. And in this way, we can save money and time by running the most important experiment, maximizing the return on investment. And in this way, we can progress the best compounds to the more expensive and downstream experiments more accurately. So to summarize the results that I've described, we've demonstrated how Cerella imputation can generate reliable predictions for peptide properties and activities, where Cerella imputation can outperform the conventional QSAR methods using 2D startup molecular descriptors, even when we have limited experimental data. And that's because it leverages the importance, relationships, and associations between experimental endpoints. Cerella can also help selecting peptides with the highest probability of meeting the desired criteria. We've shown how we have a strong correlation between the estimated uncertainty and observed accuracy, which when we combine these robust estimated uncertainties with accurate predictions and the probabilistic scoring in StarDrop, we can identify peptidic compounds with higher chances of success. Cerella can also guide the design of peptides and prioritization of experimental resources by providing insights into optimizing peptide structure, where we can look at which monomer properties and positions are important in the sequence alignment for the endpoints of interest. It can also guide the experimentation resources by focusing on the most valuable measurements to run with which to accurately impute late-stage studies. So I hope these results got you excited about Cerella, and I'm going to hand now to Matt, who is going to talk to you more about our platform.
Thank you very much, Samar. It was a really fascinating presentation and some really exciting results in a particularly challenging area. I was just really going to add just one or two words about our Cerella platform, because while it's very important to have a powerful algorithm that generates excellent results, it's another thing to be able to actually use it in practice. And so we've been really delighted to deploy the Cerella platform, customized to work in this way for these peptidic compounds to our collaborators at MSD. So I just thought I'd say a few words about what Cerella can do. And it's really about enabling sort of this active learning process in drug discovery. It automatically updates and prepares experimental data for model building. So it connects with data sources, and it applies all of those very important but tedious and time-consuming steps to clean and prepare that data, transform it into the appropriate units to get the best model performance. And it populates the sparse matrix that Samar has been describing in her presentation. And then, using the Alchemite method we've been talking about, it will build models of that data, and those models will be updated as new data become available. So you're always working with results based on the latest information, and removing that sort of burden of manually updating, rebuilding, and revalidating models every time new data become available. And of course, with Alchemite, you can fill in the blanks, again, as Samar nicely demonstrated, with these purple squares, these imputed values with the high level of accuracy and with the confidence estimate in each individual prediction. Now, this can bring a new challenge, which is if only a few % of the possible data has been measured in practice, filling in the other, 98, 99% of that data can generate very large matrices of data, potentially tens of billions of data points. So Cerella scales to manage what we call the massive matrix of imputed results, to make it very easy to query and highlight these important conclusions and results that Samar has been showing to you. And then make those very easily accessible directly on the desktops of the key decision makers in projects via our StarDrop software. Again, Samar mentioned the multi-parameter optimization approach in StarDrop. But the same results, the same conclusions, the same data and models are available through any platform that you wish via a RESTful API, making it really easy to integrate with other platforms for analysis and visualization of those data. So it's not just about the algorithms, but it's also about making those easily accessible and so that you can use those results in practice to guide drug discovery projects. So that's just the next step in the chain, if you like. And with that, I think we're ready to move on to the Q&A. So it's over to you, Catherine.
Fab, thanks so much for both of you for excellent presentation. We've got quite a few questions coming in, but just a reminder, the Q&A button's at the bottom and you can hit that whenever you have a question that you'd like to ask. Dan, it would be great if we could kick things off with a question for you, if you're there. Just about the impact of this research project on peptide discovery. What in your view is the potential impact of this research?
Looks like I should be unmuted. Yeah, so yeah, it's a great question. I mean, I think part of it is obvious in the fact that when you can get a better predictor, you can make better decisions. But really, there's a couple different aspects to this approach that are advantageous. I think that the biggest one is the fact that since we are doing imputation, we are using all of the available data rather than a standard Q-star model that would only look at only look at the descriptors itself and not actually learning from different experiments. So it gives a big advantage to the predictive power of this method. But as Samar mentioned, having the importance matrix, we can kind of understand what things are influencing, things that can help us with design, both in terms of parts of the compound or which experiments help us predict other experiments and kind of help us do some sort of prioritization in terms of both compounds and even our experimental strategy.
That's great. Thanks so much. I've got a follow-up question for you linked to that. So we've talked about peptides here, but can you discuss a little bit further the peptide endpoints that you've kind of been focusing on here, obviously in as much detail as you can?
Yeah, so I don't know, I can't give full detail on all the endpoints, but it's I would say it's all the endpoints that you would expect from biophysical assays to PK and ADME, things like solubility, etc. And it would actually depends on each project would have a different kind of suite of assays that would be that would be running that project, and we were using the actual assays that we ran on the projects.
Great, thanks, Dan. Samar, I think we got some questions for you around whether we looked at 3D descriptors at all.
Yeah, that's a very good question. So we understand that the properties and activities of peptides rely on the 3D conformations, and we've explored some of the 3D descriptors. So we've looked at 3D graph-based descriptors. We've also explored 3D pharmaco-based descriptors. However, we couldn't demonstrate the advantage of adding those descriptors, because if we are using a single conformation at a time, which it's difficult for identifying a conformation that would be ideal for a particular endpoint. So we have to either take an average or just go with the conformations with the lower energy. So yeah, in summary, we've tried them, but they didn't seem to give advantage over the conventional molecular descriptors.
Thanks, Samar. Linked to best descriptors, are there any particular descriptors that were particularly helpful for peptide activity or property prediction? Well, I guess you've kind of answered that one already. Sorry, next question. The methodology presented today, is it helpful for predicting the correct pose of a peptide when bound to a receptor? Matt, could you comment on that one?
Yeah, be happy to. So the answer to that is not this method. So this method is not particularly relevant for predicting bound poses or conformations. However, it's actually quite a nice lead up for our next webinar, where we're actually talking about predicting non-cognate binding poses in structure-based design, and actually demonstrating the application of our biopharmics platform and the Surflex doc method to that. That, as Catherine mentioned at the beginning, is actually a summary of a publication that just appeared in Journal of Computer-Aided Molecular Design, and shows that provided you have really high quality and robust conformation generation, such as the force gen method in biopharmics, you can really achieve remarkably good results for predicting bound poses of macrocycles. And so, yeah, thank you, Catherine. You brought the slide up there. I'd suggest if that's of interest to you, then yeah, please do come along to our next webinar in just a few weeks.
Thanks, Matt. Another question that you might be able to answer. If you are generating a set of descriptors for each amino acid, do you end up with far more descriptors than data points? And does this cause any problems with overfitting?
Yeah, that's a good question. So yeah, so we do have more descriptors than endpoints, but that did not have a negative impact on the predictive power of the method. We've tried to focus on the a subset of a small vector of interpretable descriptors for the monomers. And it's worth mentioning that they are not necessarily amino acids. It also includes non-natural amino acid or non-natural monomers. Yeah, so the method can handle these high dimensional inputs.
That's great. And do you have any other comments on the length of peptides on which the method works? Is there a limit?
So for the current project, we've applied peptides that are 6 to 15 monomers, but I don't see that it won't work on larger sets. So we found that it's that the molecular descriptors combined with sequence-based descriptors work very well to generate both accurate and interpretable results.
Fab. A question for you, Dan, is around the molecular modeling tools that you use to determine the best conformation of selected peptides before filling in experimental data. Is there anything that you're able to comment around that?
I think Samar can probably comment on as she was mentioning about the three-dimensional descriptors, but I'll just mention that another option that we have is just trying to adapt the suite of peptides to the for example, if we have an existing crystal conformation, we could effectively do a homology model of the peptide to the crystal conformation. But maybe Samar could talk about some of the other methods.
So in generating the 3D descriptors, we've used the conformations that you supplied to us, Dan, where we looked at, one is the graph-based method, 3D graph-based methods, where we looked at the distances between, like a connectivity map between the monomers in their 3D dimensions. We've also looked at pharmacophore-based descriptors, so looking at the distances between functional groups, and that's how we determined or described those peptides in a 3D way.
Thanks, Mar. For your sequence-based descriptors, how do you handle peptides of different lengths?
So the first step before generating or calculating the monomer descriptors is to perform peptide alignments. So for each, so even if they have different length, we would, Dan, you can comment more on that, on how specifically you've ran the alignments, but it's possible to align peptides even if they have different lengths. What matters to us is that we can identify for each monomer in the alignment specific position that we can say, okay, this is the third position in the alignment, this is the fourth position in the alignment, so that we can generate a matrix where we described each of the monomer in the positions with their vector of descriptors.
If I can just add, I think another key point here, and one of the beauties of the method we're using, is it can handle incomplete columns of descriptors. So if you obviously are aligning peptides at different lengths, there may be gaps in that alignment, or the alignments may actually start with sort of empty monomer positions. And so actually, because the imputation method can handle situations where there are missing descriptors completely seamlessly, we can actually represent that just as a gap in the descriptor sequence.